So, in our last lecture, we have finished here production of vaccine. So, irrespective of its vaccine based on a virus or a bacteria, selection of strain is very important for the production of any kind of vaccine. So, first we have to select the strain of our importance, and that particular strain should be cultured. If it is a bacteria, then we can culture it in either semi-synthetic or synthetic culture medium. If it is a virus, then we cannot culture it in synthetic or semi-synthetic medium because virus culture needs host cell. So we have to culture the animal cell, which is the host of that particular virus, and then we have to infect or inoculate the virus to the host cell culture so that the virus can grow within the cultured cell. So this portion is known as upstream processing. When we can feel that sufficient amount of growth of the microorganism is there, then we have to go for downstream processing. Means we have to go for isolation of the protein. If we are isolating the protein of the microorganism that is responsible for the antigenic property, or else if we are going for the full microbes isolation, then we have to isolate the full microbes, and in that particular case we have to go for one more step, that is known as inactivation of microorganism. So inactivation of microorganism follows different methods. So it depends on the type of microorganism. Here I have tabulated here the modes or the methods of inactivation of the virus particle. Basically, three compounds, beta-propionolactone, butyraldehyde, and formaldehyde, are used for the activation of viruses. But not all the times we can use all sorts of inactivating agent. So for the preparation of vaccine, taking a rabies virus and influenza virus, we have to add beta-propionolactone. And for the preparation of vaccine from polio and influenza, hepatitis A, we have to use formaldehyde as an inactivating agent. Whereas glutaraldehyde is used to inactivate toxins contained in acellular pertussis vaccines. So inactivation of the virus or the bacteria is a step here. Then we have, if we are isolating the protein, so then we have to centrifuge to pellet down the proteins or else the cell debris, and the proteins will be collected at the supernatant. The supernatant will be filtered, or else after filtration we can go for chromatography process. So here in chromatography process we can go either gel filtration chromatography and then affinity chromatography to purify the protein component. After purification of the protein component, we have to go for formulation. There we have to add adjuvant, then stabilizer, and the preservative. And after formulating the vaccine, we have to check for the immunogenic property or immunogenicity of the prepared vaccine. And if it is found okay, then we can go for bottling as well as labeling and lot release. This process is known as quality control and lot release. So this is a brief flow of vaccine preparation. Now we are coming into a different type of vaccine which is known as recombinant vaccine. So recombinant vaccines are produced using recombinant DNA technology, or using the technique known as genetic engineering, where genes for the desired antigen of a microorganism, it may be a virus or bacteria, uh, are, uh, are, are, are inserted into a plasmid. The plasmid should be called as a vector. Different strategies are taken here for the production of recombinant vaccines, uh, either by using an engineered vector, say vaccinia virus for vaccinia virus, or else introduction of a mutation by addition or deletion of a portion of DNA. So these are the strategies has been taken for the production of recombinant vaccines. So this picture will tell you how recombinant vaccines can be produced. So first we have to select the pathogen, and then we have to identify the proteins that is responsible for the uniqueness of the antigen property of the pathogen. Now we have to select the gene that is actually encoding this unique antigen or the protein. So this gene will be selected and cut out from the bacterial genome, from the virus or the bacterial genome. And then this isolated gene that codes for the specific antigen will then integrate or like it via restriction in the nucleus, by the function of the restriction in the nucleus into plasmid. So you know that plasmid is the extra chromosomal gene of a bacteria, and here the bacteria we have chosen, the bacteria definitely a non-pathogenic bacteria. So what we are using? We are using the bacterial plasmid as a vehicle or as a vector. Now the bacteria will reproduce, and we can get with the reproduction of the bacteria, we will get sufficient amount of vector recombinant vaccine. And that vector means the plasmid that is multiplying within the culture. So that vector contains a vaccine or the antigen gene. So this is the subunit antigen we can prepare, and this subunit antigen of the recombinant DNA vaccine can be injected into an individual for checking. In the second mode, we can also transfect the gene uh, into a genetically modified plant. But here, this transfection is not an easy way. We have to use a new method that is known as gene gun technique. So here, a gun type of instrument is used, but the vehicle is the nanoparticles, and these nanoparticles are either silver or tungsten nanoparticles, having the size is around 20 to 30 nanometer. So first we have to prepare this nanoparticle of either silver or tungsten. Then these nanoparticles will be wrapped by 100 to thousands molecules of short DNA sequences or the gene, and this gene encoded, encapsulated silver or tungsten nanoparticle will be shotgun by using this gene gun method, and this will direct the plant cell nucleus. So we can directly target the plant cell nucleus and inject the gene into the nucleus of the plant cell. So now this gene will extrapolate there, will encode the protein, so we can get the protein via ingestion of the GM plant, and we can get the vaccine within ourselves. So this is not that the method is not like we have to inject subcutaneously or intramuscularly. The third method is a modified method, is a modified for expression in the host, and the DNA is injected, DNA vaccine is injected uh, into the host via this gene item. So this is the method for the preparation of recombinant DNA vaccine. Now we are coming into a particular section, different section, that is DNA vaccine. So DNA, what are DNA vaccines? Vaccines that contain DNA that codes for the specific protein, means the antigen from the pathogen. So we are taking, isolating the DNA, and the DNA is injected into the host cell, and this DNA, after entering into the host cell, uses the DNA to synthesize a protein, and this protein has the antigenic behavior or antigenic property. So technique for protecting an organism against disease by injecting it with genetically engineered DNA to produce an immunological response within the host. So this is the definition of DNA vaccine. Now, nucleic acid vaccines or DNA vaccines are experimentally proved and applied to a number of viral, bacterial, and parasitic diseases. And also, nowadays, scientists are trying these DNA vaccines for the treatment of different type of tumors and cancer. So, they are applying it in tumor model also. Now, the question is how this DNA vaccine works, or else the mechanism of DNA vaccine. A DNA vaccine intended to uh, induce an adaptive immune response or acquired immune response needs to encode an antigen as well as an adjuvant. So the according plasmid DNA, here you can see this is the plasmid DNA, pDNA, plasmid DNA is applied either systematically or topically. That is by intramolecular injection, intramuscular injection, and transfected. This will transfect this DNA after entering into the muscle cell or else into the skin cell. So they transfect within the keratinocytes or the myocytes, and expresses transgene and release derived peptide or else the protein via exosomes or apoptotic bodies. So here you can see this DNA is transfected here either in keratinocytes or else in myocytes, that means muscle cells. Now they will encode some proteins. This red one is at the proteins, and the proteins are coming out by the process by the vesicles are called exosomes or else in the form of apoptotic body. So this material is endocytosed by immature dendritic cells. Here you can see they are endocytosed by this immature IDC, immature dendritic cell. So they have this IDC have endocytosed this antigen or the protein, which subsequently uh, present antigen preferentially via major histocompatibility complex type 2 molecules and interact with the CD4 T cells, that is the helper T cell, in draining lymph nodes. So now they are coming here to the lymph node. Okay, from vessels, blood vessels to the lymph node. So direct transfection of antigen-presenting cells, including the immature dendritic cells, results in endogenous transgene expression and hence parallel presentation via MHC, MHC. So parallel presentation here, parallel presentation via this APCs by the two type of MHC molecule, MHC1 and MHC2, yielding both, sorry, yielding both CD8 plus means cytotoxic T cell as well as helper T cell. So that response goes in parallel. And besides this cellular immune response, a humoral immune response is induced. Here is induced by the CD4 T cell to the B cell, and now the B cell will release antibody, will produce antibody. Means here the plasma cells are producing the antibody. So here the humoral immune response is induced if the B cell receptor recognizes the protein antigen and occurs helped by the pre-activated antigen specific CD4 T cells or the helper T cell. So this is the mechanism how DNA vaccines work. Now there is a difference between recombinant DNA vaccine and simple DNA vaccine. So DNA vaccines are vaccines which use fragments of DNA, whereas recombinant vaccines are vaccines that carry a recombinant DNA molecule or a vector. So the form of vaccination is DNA in case of DNA vaccine, and in case of recombinant vaccine, this is the recombinant vector or else the recombinant organism. So here in DNA vaccine, we are not applying genetic engineering or recombinant DNA technology, but to prepare recombinant vaccine, we must use recombinant DNA technology or else genetic engineering. So this is a brief difference between the two types of vaccines.